Yes, so the second video of ours is on the streptococci. Streptococci. Okay. So streptococci. What is streptococci? Strep means in arranged in the parallel manner like this, and cocci means round. So it is round cocci. Okay. So classification. If I want you to classify this on the basis of O2, there are two classes, aerobic and obligate. In aerobic, we have aerobic and facultative aerobic organism and facultative anaerobes. Facultative A and EU aerobes, ठीक है. And in second we have obligate anaerobes. A and E anaerobes. In this mainly we don't classify it as more because we have only one category, which is streptococcus. Streptococcus. लिसन मैंने वीडियो मत मीन्स इसलिए बनाई है जिसमें आपकी फाइनल एग्जाम की मेन तैयारी हो सके. ठीक है, तो मैंने इस क्लास के हिसाब से नहीं बनाई है, इंपॉर्टेंट जो मुझे लगा उसके हिसाब से बनाई. ओके. सो ऐसा मत बोलना कि मतलब कम बताया है या ज़्यादा बताया. मैंने आखिरी में, क्योंकि बहुत कंफ्यूजिंग हो जाता है, इसलिए उस बेसिस पे मैंने करवाया. ओके. सो इन द aerobic we classify it on the basis of hemolysis. In hemolysis we all know that there are three types of. First is alpha, second is beta, third is gamma. First, alpha hemolysis is in the blood, which shows green hemolysis. It is a partial hemolysis, and in the beta hemolysis it is complete hemolysis, shows yellow color. And in gamma hemolysis we shown, actually we don't have any hemolysis here. It's a recording or not here? No hemolysis. Mainly example enterococcus group. So beta, it is the most important one. And in serological typing, on the basis of carbohydrate, see, we divide it into the 20 lanes field group. 20 lanes field group. You have many questions with this classification, like which of the organisms show Lancefield grouping. So the answer is streptococci. Okay. So in Lancefield grouping we have groups from A to B alphabets except I and J. Okay. Example is the group A beta hemolytic bacteria streptococci, which is pyogens. And you come to know in the pathology that it is the most important organism for the this rheumatic heart valve disease, rheumatic fever disease. It is the beta hemolytic disease, beta hemolytic streptococci pyogens, which show pharyngitis in children with heart valve diseases. Okay. So it is the important one. Then in this only the second classification is in the Griffith typing. Griffith type, in which we have 1, 2, 3 up to 150. So these are the two types of major classification of this. So next, if we go to the introduction, like in the previous video I told about that I will give responses according to introduction, morphology, culture, biochemical reaction, this. So the second one we have morpho. So I already told you about this, that it is a chain like, arranged in a chain like manner, and it is a cocci chain, non-motile, except some are motile. Non-motile is streptococcus pyogens. Okay. But all other are motile. And non-sporing. These are the main morphological characteristics, estimations. Then third point is our culture. Normally we can grow it in the solid media, and if we want to grow it in the if we want to grow it as very fast, so we can grow it in the blood agar, or we can also grow it in the sugar agar or serum agar. Okay. Biochemical reaction, it is a catalase negative. In the previous video I also told about this. We can distinguish staphylococcus from streptococcus by the catalase positive or negativity. So it is a catalase negativity, and it gives PYR test. Okay, it is an important test for streptococcus pyogenes to differentiate from other streptococci. It is a PY test is a pyrimidinase test. It is something like that only. Streptococcus pyogenes, it is used for distinguish from other streptococci. Then also it is a bile insoluble. Okay, it is a bile insoluble, so it is positive for bile solubility test. Means it is negative for bile solubility and positive for bile insolubility. Okay, and in the, if we want to distinguish this from pneumococcus, streptococcus pneumococcus, then we can use this test as bile insolubility test, where you can see that streptococci and pneumococcus is bile soluble. Okay, so we can differentiate in this. Resistance, next point. Next point is resistance. So in resistance you can see that this organism pyogenes is mainly tolerable up to 56 degrees Celsius of temperature for 30 minutes. After that only it is killed. And its capsule contain hyaluronic acid, which protect it from the severe temperature, and also it contains crystal violet. That's why streptococci can survive in this, and staphylococci, which is grapes like, is killed in crystal violet. And it also contains cell wall protein for myocardium. Okay, then the next point, what color you want? Okay, dark blue. Next point, we discuss on antigenic structure. In antigenic structure there are majorly three: capsule, carbohydrate C antigen, and the third is surface protein. Surface protein are the protein which are different types of protein like MTR, and it mimics the cells of human body. Okay, uh, it mimics the cells of human body. Like I give you example from the pathology in the rheumatic heart valve disease, rheumatic heart fever. The bacteria, this streptococcal M protein will mimic like myocardium or heart valve cells. So it cannot detect by the normal physiology of the human body or normal defense mechanism of the human body. Okay, so these are the important surface protein, they actually mimic. Then if we take capsule, it contains capsular hyaluronidase acid present in human synovial fluid, or it mimics as human synovial fluid. Then group A carbohydrate in the cardiac heart valves, then cytoplasmic microbe antigen as vascular intima, it behaves like that, and the peptidoglycan, it behaves as skin antigen. Okay, then the next, इसका भी कलर चेंज करते हैं. अरे कलर चेंज हो भाई, क्या कर रहा है. Violet. Toxins. Toxin, there are majorly the hemolysin is present. Hemolysin. What is the special property of hemolysin? So you can see here there are two type of lysins, like streptolysin O and streptolysin S. Streptolysin O is the O level, which is oxygen level, and it is O stable. Then it is heat level and not work, like no work in O2 की absence में, not able to work in the incapable of working in the absence of O2. And the second one, which is the streptolysin S, which is the O stable, it is heat resistance, okay, and work well in O2. This is the main thing. Then for diagnosis, like it is the point of pathology, but you can also make clear this point, that is the streptolysin O, this make antibody against body, like it make antibody first of all against this. And in the diagnosis we can, in the मतलब like जब आप मतलब वो देखोगे, टाइटर देखोगे ना इसका, तो आपको प्रेजेंस ऑफ एएसओ टाइटर दिखेगा. और एएसओ टाइटर मतलब एंटी स्ट्रेप्टोलाइसिस ओ टाइटर में आप इसको पहचान सकते हो कि ये ऑर्गेनिज्म प्रेजेंट है या नहीं. लाइक एएसओ टाइटर फॉर दिस टेस्ट, एएसओ टेस्ट इसको बोलते हैं. और एएसओ टाइटर में अगर 200 से ज़्यादा मिलता है तो एएसओ टाइटर आपका पॉजिटिव आता है. ओके, और ये मेनली ये रूमेटिक हार्ट डिवेल्टिस इन सब में चेक होता है एएसओ टाइटर, उनका डायग्नोसिस लैब टेस्ट है कि मतलब प्रेजेंट है ह्यूमन बॉडी में कि नहीं. ओके, सो नेक्स्ट पॉइंट, एंजाइम्स. First is exotoxin. Enterotoxin it for what? For food poisoning. And exotoxin for rash, it promotes rash. Then enzymes में exotoxin, it promotes rash, well, means it is a type of toxin, it is not included in enzymes, it is a toxin. Then in enzymes we have three type of enzymes, like streptokinase, deoxyribonuclease, and third is hyaluronidase. These are the three one. Pathogenesis. In the pathogenesis you can see that it promotes respiratory infection, tonsillitis, and pharyngitis. Another is the skin infection, wound and burn. And the non-pyogenic infections. Majorly the non-pyogenic infections are acute glomerulonephritis. It is the inflammation of the basement membrane of the kidney. You can read in the third year in the pathology, because in Russia it is in the third year. And the second is the acute rheumatic, which I already told you, acute rheumatic fever. Along with this, it causes the heart valves, inflammation of heart valves. Then lab diagnosis. कितना मिनट हो गया? Okay. In the lab diagnosis, you can see that first we have to collect the specimen from pus, swab test, or blood. Okay, then collection or transportation of the media. So we can take pike transport media. Pike transport media, and it is important. Like if you have the मीन्स अगर आपके पास ये है, आपने जैसे निकाल लिया कोई भी लैब डायग्नोसिस स्पेसिमेन निकाला, अगर आपको कोई दूर जगह आपको ट्रांसपोर्ट करना है या माइक्रोबायोलॉजी लैब वहाँ है नहीं, तो आप इसको पाइक ट्रांसपोर्ट मीडिया में रखोगे जिससे कि ये सेफ रहे ट्रांसपोर्टेशन के लिए। ओके, सो थर्ड ग्राम स्टेन करके हम इसको देखेंगे, चेन लाइक मिलेंगे। फोर्थ इज कल्चर ब्लड अगार में। फिफ्थ वन इज सिलेक्टिव मीडिया, इसका CVBA मीडिया, क्रिस्टल वायलेट ब्लड अगार, जो मैंने आपको बताया क्रिस्टल वायलेट ब्लड अगार। और ये कि कुछ इम्पोर्टेंट में से इसको याद रखना हरदम, क्रिस्टल वायलेट ब्लड अगार और पाइक ट्रांसपोर्ट मीडिया इसके लिए, ठीक है। देन इसका सिलेक्टिव मीडिया, मतलब यही इसका सिलेक्टिव मीडिया था CVBA। एंड अलॉन्ग विद दिस इसको आप PNF में भी रख सकते हो, ये भी आप याद ही रखो एक तरीके से। PNF आपका ये है कि पॉलीमाइसिन B, नियोमाइसिन एंड फ्यूसिनिक एसिड मीडियम, फ्यूसिनिक एसिड मीडियम में। कॉलोनीज कैसी दिखेंगी, इम्पॉर्टेंट है ये पहचानना। कॉलोनीज किस हिसाब की दिखेंगी आपको, अगर स्मॉल, मेजरली इसकी स्मॉल कॉलोनीज बनती है विथ लो कॉन्वेक्स। इफ दे फॉर्म मैट लाइक then it is virulent. Okay, if they form glossy shiny colonies then they are avirulent. And for motility, if they are motile, you can use hanging drop method, which you all know in your general microbiology. Thank you for this video. Okay.